Hello and welcome back. So in this video we're going to go over now a dihybrid cross. So with a dihybrid cross we are going to track two traits. Okay, the two traits that we're going to track now are going to be seed shape. Okay, we're looking at either round or wrinkled peas. Okay. And we're going to look at color as our second trait. So we've either got yellow or we've got green. All right. Now you should be able to, or I'm going to expect you to be able to, identify the dominant versus the recessive trait if I give you the letter. Okay? Because remember, our dominant trait is always going to be the capital letter. Our recessive trait will be the lowercase letter. Okay? So in this case, our dominant is going to be round and yellow. Our recessive traits are going to be wrinkled and green. Okay? And if you notice, the letters are easy to distinguish: capital versus lowercase, capital versus lowercase. Okay? That's the other thing you want to make sure with your crosses. All right? And what the dihybrid cross is going to explain is Mendel's second law. And Mendel's second law is going to cover independent assortment. Okay? One thing to keep in mind with Mendel's second law, it only holds true for genes that are unlinked. If you've got linked genes, things get more complicated. Again, we're not going to worry about following linked genes in this class. If you take a genetics class, that would be something that would be part of the curriculum. For this, what I want you to just keep in mind is the traits we're following have to be unlinked, meaning the traits that we're following are on separate chromosomes or they're far enough apart on the same chromosome that they don't behave as linked because of the crossing over that happens during meiosis. Okay? What the law of independent assortment gets to, okay, is that different traits segregate to the gametes independent of one another. Okay? So what we're going to do to follow Mendel's second law, all right, we are going to follow traits that segregate independently, okay? And the way we're going to map that out is, again, using our Punnett square. And if you notice, our Punnett square now has gotten more complicated. I've drawn it up here ahead of time so you don't have to listen to my squeaky marker, although some of these markers are still going to squeak. All right? What we're going to do for our dihybrid cross, okay, we're going to follow two traits, and the example I'm going to use, our parental generation, is going to be heterozygous at each locus. Okay? So mom is going to be heterozygous for seed shape. Mom is going to be heterozygous for seed color. Dad is also going to be heterozygous for both traits. Okay? That's also one of the things with nomenclature and genetics. It's always ladies first. So whenever you see something set up as a cross like that, okay, we're crossing a heterozygous with a heterozygous. The first one is typically the female. Second one is the male's gametes. In this case, they're both the same. They aren't always going to be, all right? And you're going to see some different ratios come out when you change the genotype, okay? But for now, what we're going to do is we're going to go through this using the parent, okay? And our parent is going to be heterozygous for each locus. And you want to keep the two letters together, okay? So you want to keep the R's together and the Y's together in this example, all right? And what we're going to do And the way that we're going to assign the gametes is using the FOIL method. Because really what I've found typically with students is the hardest part they have is assigning where the gametes go on next to the boxes here. All right? So if you remember back to your math class and the FOIL method, that's what we're going to use now to assign our gametes. Okay? So the F is going to stand for first. Okay? So the way we're going to do this, we have to treat each of these traits as independent. So with our gametes, we have to have one R and we have to have one Y. Okay? So what we're going to do, we're going to take our first two letters, capital R, capital Y. That's our first combination of gametes. Okay? So we're going to put that on our Punnett square. All right? Next, the O stands for outer. So you're going to take the outsides. Okay? So we've got our capital R, our lowercase y. That's our next combination of gametes that can go into our Punnett square. Capital R, lowercase y. Let me loop my y just for consistency's sake. Okay. The I now is going to be inner. So we're going to take the inside two. Okay. So the lowercase r, the capital Y. Lowercase r, capital Y. That's our next set of gametes. And then finally, or last, we're going to take the last two. We've got lowercase r, lowercase y, okay, in terms of our traits. Now, because mom and dad have the same genotype, dad's gametes and mom's gametes are going to line up the same, okay? So we would fill in our vertical set of boxes with the same letter combinations we have up top. So we've got capital R, capital Y, capital R, lowercase y, green, we need green, lowercase r, capital Y. This one doesn't look very green. Okay. And then finally, lowercase r, lowercase y. All right. What we're going to do then is we're going to fill in our Punnett square based on the genotypes that we've assigned for our gametes. Okay. So in this case, with our first box, we've got two capital R's. You want to keep the R's together. We've got two capital Y's. Okay. And if we go down our boxes, capital R, capital R, capital Y, lowercase y. This now we've got capital R, lowercase r. Recessive trait always comes second. Okay. Capital Y, capital Y. All right. And I'm going to go ahead and fill in the rest of our Punnett square here, and this will take me a minute or two. Okay. I do have an example in the PowerPoints using eye color, and I think height as the two traits. Okay. So if you want to practice doing a Punnett square with the exercises I have in the PowerPoint, that would be highly recommended. Okay. We'll also have either some homework problems or some practice problems for you so that you can practice doing this. Because really, the more you practice doing these, the better you're going to get at it. If you're taking the lab class, this will be something that you're going to go over in lab. Okay. Little r, capital Y, lowercase y, capital R, little r, uppercase, lowercase, capital R, little y, little r, capital Y, little y, little r, little r, little y, little y. Okay? So that fills in our Punnett square. And what we're going to get out of our Punnett square when both parents are heterozygous. Now this is only true when both parents are heterozygous. We are going to have a phenotype ratio of 9 to 3 to 3 to 1. Okay? So this is now going to require you to interpret what we just did. All right? This is something else I'm going to expect you to be able to do. Okay? And what we're going to do is we're going to look at our Punnett square and figure out the phenotypes. Okay? We'll start with this first one. Okay? And I'll use a yellow marker, because the P for this particular genotype would be round and yellow. Okay? Anytime you have the dominant allele, that's going to be round. The dominant Y is going to be yellow. So you want to look for all the dominant R, dominant Y combinations. And I'm going to underline those so that you can see them a little better in looking at these. More. Okay. Now, if I've filled this out correctly and I've tagged all of them, hopefully I've done that, okay, what we're going to see is 1, 2, 3, 4, 5, 6, 7, 8, 9 potential genotype combinations that will give us a round seed and a yellow P. Okay? So that's where our 9 comes from. All right? Next, we'll look at, with our phenotypes, we are going to have 3 that are going to be round and green. And I'll underline those in pink so you can see those. Okay? So round and green, we've got 3 different genotypes. Now if you notice, you've got some different genotypes that will give the same phenotype. It can be homozygous dominant for seed shape, but in order to have the green color, it has to be homozygous recessive for the color trait. Okay? So the little y's are going to give us the green, big R is going to give us the round. If you notice that with our 9 round and yellow, not all of them have the same genotype. I'm not so worried about you following the genotypes on this because we've got a lot of different genotypes. What I want you to take away is the 9 to 3 to 3 to 1 ratio with phenotype. Okay? So our next one, let's do in blue, just for fun. Okay? So next we're going to have seeds that will be wrinkled because they're homozygous recessive. And our seed color is going to be yellow. Okay? So we'll have 3 that are going to be wrinkled and yellow. Okay? Our last one, we'll use the orange marker, okay, is going to be the least common. It's in this box over here, okay? And I can't write any lower on this, so I'm going to write it over here. The one that you're going to get is going to be wrinkled and green. Okay? So from the information we set up in our Punnett square, we can figure out the genotypes that are possible. We can also figure out the phenotypes. Okay? In this case, you've got a lot of different genotypic combinations because we've got 2 traits. So what I want you to concentrate on this one is the phenotype ratio. Okay? I've got another example in the PowerPoints you can practice with, but those dihybrid crosses, that's going to be how those work. Okay? Hopefully that makes sense. If you have any questions, please let me know.